Glycolysis is an anaerobic process that converts glucose to pyruvate, yielding only a small portion of the ATP available for glucose. The majority of ATP produced during glucose metabolism comes from aerobic processes, which begins with the complete oxidation of glucose derivatives to carbon dioxide. This oxidation occurs through a series of reactions known as citric acid cycle or the tricarboxylic acid TCA cycle, commonly referred to as Krebs cycle. The citric acid cycle serves as the final common pathway for oxidizing various fuel molecules, including carbohydrates, fatty acids, and amino acids. Most of these molecules enter the cycle as acetyl coenzyme A. Under aerobic conditions, pyruvate generated from glucose is oxidatively decarboxylated to form acetyl coenzyme A. In eukaryotic cells, the citric acid cycle reactions occur within the mitochondria, while glycolysis, mitochondrial specifically the citric acid cycle reactions occurs within the mitochondrial matrix, while the glycolysis takes place in the cytoplasm. The citric acid cycle serves as the central metabolic hub of the cell, acting as a gateway for the aerobic metabolism of any molecule that can be converted into an acetyl group or a component of the cycle. Additionally, it provides crucial precursors for the synthesis of various other molecules, including amino acids, nucleotide bases, and porphyrins, which are the organic component of heme. Furthermore, oxaloacetate, a key component of the citric acid cycle, plays an important role as a precursor for glucose synthesis. The citric acid cycle plays a crucial role in converting fuel molecules into ATP by facilitating oxidation of carbon compounds that can lose electrons. This cycle involves a series of oxidation reduction reactions during which an acetyl group is oxidized to produce two molecules of carbon dioxide. This process generates high energy electrons, which are subsequently utilized to drive ATP synthesis. Essentially, the primary function of the citric acid cycle is to harvest these high energy electrons from carbon based fuels. The overall structure of the citric acid cycle is that it begins with a 4-carbon compound, oxaloacetate, which combines with a 2-carbon compound having acetyl unit to form 6-carbon tricarboxylic acid, and this 6-carbon compound undergoes two successive oxidative decarboxylation, hence releasing carbon dioxide and generating high energy electrons. As a result, a 4-carbon compound remains, which is then processed further to regenerate oxaloacetate, allowing the cycle to start anew. Throughout this process, 2 carbon atoms enter the cycle as an acetyl unit, and 2 carbon atoms exit in the form of 2 molecules of carbon dioxide. It's important to note that citric acid cycle does not produce a large amount of ATP and does not directly use oxygen as a reactant. Instead, it extracts electrons from acetyl coenzyme A, which are then used to produce NADH and FADH2. Specifically, 3 hydride ions, which is equivalent to 6 electrons, are transferred to 3 molecules of nicotinamide adenine dinucleotide NAD+, and 1 pair of hydrogen atoms, which means 2 electrons, is transferred to 1 molecule of flavin adenine dinucleotide FAD. When these electrons are oxidized by oxygen during oxidative phosphorylation, they can yield a total of 9 molecules of ATP. The electrons released during the reoxidation of NADH and FADH2 travel through a series of membrane proteins known as the electron transport chain, allowing a proton gradient across the membrane. This proton gradient drives protons through ATP synthase, resulting in the conversion of ADP and inorganic phosphate into ATP. The citric acid cycle, along with oxidative phosphorylation, is responsible for the majority of energy utilized by aerobic cells, accounting for over 90% in humans. This process is highly efficient as the oxidation of just a few citric acid cycle molecules can produce substantial amount of NADH and FADH2. The 4-carbon compound molecule oxaloacetate, which initiates the first step of the cycle, is regenerated at the end of each complete cycle. This means that a single molecule of oxaloacetate can participate in the oxidation of numerous acetyl groups, acetyl molecules, maximizing energy production. Thank you.